to empower clinicians, researchers, and healthcare professionals with cutting-edge knowledge and actionable insights from the world of cancer genomics. As genomics, we know, is reshaping quickly the cancer care globally, we believe it is essential to demystify genomic science and enable its integration into everyday clinical and research practice in India as well. Despite the explosion in genomic technologies, from next-generation sequencing to multi-omics, there remains a gap between scientific innovation and its clinical application. So over the next 50 minutes or so, we will try to introduce some key concepts and terminology in cancer genomics, understand how genomics informs diagnosis, treatment, and patient prognosis in our hospitals and clinics. And we would also try to explore some of the new exciting tools that are currently widely open for us to use. We are honored today to have a panel of distinguished speakers joining us this evening. We have Professor Arvind Krishnamurthy. He is the professor and head of surgical oncology at Cancer Institute, Chennai. A widely respected voice in oncology, Professor Krishnamurthy brings rich clinical insights and a strong translational perspective to this conversation. We have Dr. Kriti Kaushik, who is the product manager at Premas Life Sciences. Dr. Kaushik will walk us through multi-omics to offer a 360-degree view of cancer genomics status in India. And we have with us Dr. Abhishek Chaudhary, who is a business development manager at Premas Life Sciences. He would talk us about the real-life challenges and some of the techniques which are in conjunction with genomics to predict better prognosis and diagnosis. So, Kriti, I would like to start off with you.
Yeah, hi, Ankita. Hi.
Hi.
And thank you so much. So I will share my screen.
Sure. So Kriti, from you, we would like to understand that what exactly does genomics mean in context of cancer care? And
sure.
Maybe you shed a light on us and...
Thank you so much. Definitely, I'll try to do justice to the topic given today. And I hope you can see my slide.
Yes, we can.
Okay. So first of all, thanks for inviting me for an insightful discussion here and letting me talk over a topic which is very close to my heart. Because over the years, I have been a part of oncology research for 8 years during my academic career at AIIMS Delhi. And the subject is so vast that every time you happen to investigate more into it, you deep dive more into it, it is always lesser. And I just want to share a personal experience where one day I was walking through the oncology OPD to meet one of my clinical collaborators. And there I encountered 2 patients who were sitting down with their report. And they were trying to understand, basically, they were diagnosed with the same kind of a cancer subtype, and they were actually responding differently to the treatment. So after such a case, I encountered many such cases over the years and found that cancer, definitely we know, is a complex disease, but there is something more to cancer than just investigating the genes. So there is something beyond the genes that we should look upon to, understand the cancer's complexity from different biological angles, which I think are the different omic layers of epigenome, transcriptome, and proteomics. And that's where I headed into multi-omics. So I will just give you a brief overview. We all know this. I think we have quite profound clinicians in our session today. And we all must be aware that how cancer is it's becoming the leading cause of death, not just in US, but globally. It is the second most leading cause of death in US. But over the years, it has been seen that cancer mortality rate has gone down. And this is because there are a lot of tailored therapies which are being designed. So we all know that there are different types of cancers and they have certain universal hallmarks, but the mechanisms by which these hallmarks actually appear these are investigated to produce tailored therapies, to produce personalized therapies. And now we don't have just one standard care of treatment for every cancer, which is just the first line of treatment, which is chemotherapy or radiotherapy. Depending upon the cancer subtypes, there are different therapies available. So let us first understand that why at all should we use genomics in cancer care. So what is cancer genomics? Study of genetic mutations, alterations, or any expression patterns in cancer cells is actually cancer genomics. And why should the clinicians, first of all, use it? Because obviously to design personalized treatment, as I said, that cancer being a complex disease, we need to design targeted therapies. So also to better understand the prognosis of the disease and the risk stratification, we need clinicians to understand better cancer genomics, and obviously how the patient is going to respond to the treatment, the therapy resistance prediction through pharmacogenomics, and minimal residual disease monitoring. So for all these cases, it is required that first of all, we need to understand the cancer from the root, which is the cancer genomics. Now, how it is being used clinically today, if I give a real life example, so there are a lot of genomic tests available across some standard cancer types, such as for lung cancer, we have the known EGFR, ALK, and ROS1 genomic test, which are the very common genomic mutations. And then we have against breast cancer, we all must be aware of BRCA1, BRCA2, and HER2 type. And then we have also oncotype diagnostics. And for colorectal cancer, we have KRAS, NRAS, and BRAF. Now, what tools are generally used for determining these genomic factors? It is the most popular tool right now available is the next generation sequencing, which is a high throughput method, which can investigate the entire genome in one go and can find the mutations. And then we have some classical methods such as fluorescent in situ hybridizations, PCR-based tests, and now the upcoming liquid biopsies as well. So when actually the clinician should decide that they should go for cancer genomics testing? So it is when the treatment options are not clear, and that happens generally when the cancer is rare category, or it has it is having a young onset, it is having early onset during the lifetime of the patient, and obviously to decide what kind of a standard care that they should opt for the patient. So in that case, clinicians these days are using different tumor boards with molecular pathologists or genetic counselors, and they take the help of genetic counselors when familial risk is suspected. So how genomic insights is useful at multiple stages of patient journey. So if we see that apart from just cancer diagnosis, it is used for the early detection and diagnosis. So definitely if we do a tumor DNA profiling at an early stage, it can reveal mutations, which can define the cancer subtypes. And apart from cancer, it is also used for prenatal or newborn screening or diagnosing any rare disorder. Then once the cancer is detected, during the treatment planning, it plays an important role, because by understanding the mutations, targeted drugs can be prescribed. Pharmacogenomics can be applied, that where we can actually study the patient's response to the drugs based on the mutations. And also we can, clinicians can make a decision whether they should opt for gene therapy or not. And suitably it is checked for studying the genetic mutations. Once the treatment is planned and the patient has given the treatment, then comes the disease monitoring. Now, small traces of the disease can be detected at the genomics level. So minimal residual disease can be used to detect by genomics. And not just that, we use liquid biopsies where circulating tumor DNA analysis help to track the tumor evolution and to understand the resistance mutations and relapse in the real time. Next, once we are done with the disease monitoring step, then comes the prognosis or the risk stratification. So genetic variants also help you to inform about the disease severity, likely progression, or the risk of recurrence, enabling more proactive management. So what all genomic information a clinician can gain from the genomic testing? So we can obviously get different mutations or single nucleotide variations, which will help to better diagnose and select a particular therapy for the particular cancer. Then we also have copy number changes, which again will be used for the same. We can understand the gene expression patterns by transcriptomics or fusions or translocation patterns, which is very common in hematological cancers. And then there is this upcoming genetic mutation, or I would say epigenetic profiling, that helps to better understand the tumors or classify them into different subgroups, which is methylation. So methylation controls a lot of expression of the genes and ultimately the cancer subtypes. And obviously we can better understand pharmacogenomics and select the drugs accordingly and even dose them accordingly. So once the genomic test is performed, the report comes, and how a clinician is supposed to analyze the report plays an important role. So yes, it is not like shooting in the dark, and not that we cannot educate the clinicians about it, and it's very, I think it's getting very popular these days. So first we need to understand the variant classification, and in fact these days most of the labs which are performing the genomic tests and they prepare the report, they try to prepare the report in a much simplified way so that it becomes easier for the clinicians to understand. So every report which will be coming up will have a variant classification, which will likely classify them as pathogenic or likely pathogenic, or variant of insignificant, unknown significance, or benign or likely benign classification. Now, after this classification, we can also use the clinical summaries which come along with the report, which help in giving certain recommendations for the therapy or diagnosis. And generally, they list out an interaction table where they have a drug-gene interactions. Apart from that, a clinician can also look around some trusted online tools such as ClinVar, PharmGKB, and OncoKB. And based on these tools, many certain third-party tools are also designed with which, if they want, with the help of a bioinformatician, they can seek out help from the bioinformatician to better understand how they can actually interpret the report. And strongly recommendation is that they need to collaborate with the genetic counselors and molecular pathologists so that they can get their opinion also and can come over for a holistic opinion. Now, coming to the concept of multi-omics. So now we have understood how genomics is playing an important role and what is the role of understanding the genomics. Now, coming to the next layer, which is multi-omics, as I spoke about it previously in the beginning of my talk, that it is now coming beyond the genes. So we are just not focused onto genomics, which helps us to provide a mutational profile, but there are other certain omic layers which we need to investigate, such as the epigenomics, where we generally look for DNA methylation patterns and check for the regulation of gene expression. So they help to control the expression of genes, which generally is seen in the transcriptomic layers, where there is a complete set of RNA transcripts. This is transcriptomics and proteomics are the functional set of genes. So whatever is expressed basically comes in the transcriptomic layers. And by studying the transcriptomics, we can help to discover new therapy and cancer driver identification and even the cell population diversity. And then we have proteomics, ultimately, which is the ultimate functional layer. So if we happen to study the proteins expressed by the cancer cells, we can determine the certain biomarkers which are unique to the cancer cells and definitely design a therapy response against it. And all of these omic layers can be studied at different resolution. So we have the bulk approach where we take up all the cells together and try to take out the nucleic acid and investigate each of these omic layers. Then we have another layer, which is the single cell approach, where we actually isolate each one cell by addressing them and try to understand the omics within one single cell. And then we have the upcoming spatial technology, where just in the histopathology slide, we try to see the expression of the transcripts or the genomic layers or the proteins onto the spatial platform into a histopathology slide where they are spatially regulated. We can see that how the cells are arranged, and within those cells, how the transcript or the proteins are expressed. And then obviously we have the liquid biopsy. So as I said, that tumors are quite complicated. So it is just not the cancer cells. They are more than the cancer cells. You have different immune cells along with them. And in order to study the interaction between them, it is important to study how multiomics is playing an important role, and also at what resolution. I mean, if we keep on increasing the resolution of these multiomics, I think it will better help us to understand each one of the interactions and better characterize the disease. So multiomics enable the precision medicine because it helps us to further classify each of the cancer patients into the subtypes and then to better design the personalized therapies against them. So one such example is, for example, we all know that there are 20 to 25% of the breast cancer cases which have an overexpression of HER2 gene, and they definitely could benefit from the anti-HER2 therapy. But it is seen that under this 20 to 25% of the cases, approximately there is a variable spectrum, okay, between 20 to 60% of these HER2 enriched tumors which give a varied response to the anti-HER2 therapy. Now, why such a thing is happening is a point of concern, because every therapy, it basically produces a change in every omic layers, and we need to characterize that change, what is happening at every different omic layers, so that we can better design the variable, a personalized therapy against it. So this is where the multiomics research is going. Because of all these things, multiomics research is increasing day by day. Over the past decades, it has considerably increased, and even the funding for multiomic approaches is increasing. So I would like to give here some real life examples of how multiomics has helped to predict the cancer treatment. So here is a melanoma treatment response. So this is an example from the Long Lab at Australia Melanoma Institute, where they have used multiomics to predict melanoma response to anti-PD-L1 therapy. So we all know that the cancer cell expresses a PD-L1 receptor over the surface. And if we can, basically this PD-L1 actually signals negatively regulates the immune response. And if we can have an anti-PD-L1 therapy, this negative response will be terminated, and then it will be better detected by the immune system, and it will help in the effective treatment. So a few associated factors were studied in response to the anti-PD-L1 therapy for the melanoma treatment. So what this group hypothesized is that there is no single ohm which can predict the melanoma treatment response, but multiomics robustly predicted the response. So they have used different omic layers to understand and prepare a model to predict that which basically category of patients will undergo which kind of a response. So they studied first the genomic layer, where they tried to see the tumor mutational burden. And so tumor mutational burden means the mutations per megabases in the genome. And they found that higher the TMB, higher the tumor mutational burden, it was associated with the overall good response to the therapy given to the patients. Simultaneously, they also checked for the structural variants. So they happened to see that there was an opposite response to the structural variants. More of the structural variants were giving a poor response to the therapy. So then they checked another layer of omics, which is the transcriptomics, where they happened to see that there was a high interferon gamma response which was upregulated in good responders. And then they used another omic layers, which was epigenomics. So they performed some methylation microarray, and they found that the PSMB8 methylation in the PSMB8 gene, along with few others, was associated with a poor response. So with all this combination, they happened to form a model where they could see that basically they use these different omic layers of whole genome sequencing, RNA sequencing, immune profiling, and methylation, and found that they basically bent the good responders versus the poor responders. And they found that the poor responders were basically associated with complex structural rearrangement and PSMB8 promoter methylation. And further, they stratified them into discordant tumors, into poor response and actual good response. So this way, they basically tried to formulate a model. Then it also, there is another case study which helped to reveal the prognosis, therapeutic and diagnostic insights for meningiomas. So this is an case study from the Zaid's lab at the University of Toronto, where they first used the basic genomics and epigenomic layers to better understand the prognostic groups in a meningioma patient category. So based on the prognosis, they actually bent the entire patient group into 4 categories, depending upon their prognosis, as you can see in the ROC curve. Then they used a transcriptomic layer to understand that which kind of pathways are upregulated or downregulated. And they found that in the 4th category, which is having the poorest survival rate, they found that in this category, there was a high upregulation of histone deacetylation. So based on this, they basically hypothesized that if in this category of patients, they can inject the HDAC inhibitors, which is vorinostat. Okay, so will there be a change in the therapy response? So basically, they try to do this experiment in mice, where they injected the patient cells in mice and did this experiment. And they found that the therapy response considerably increased in the group 4 category. Then they used a proteomic layer where they tried to find out the phenotypic insights and actionable biomarkers. And they found that there were some unique protein biomarkers in each of these 4 categories of the patient subgroups divided on the basis of the prognosis. And it has basically simplified the diagnosis, this entire research, because now based on just these protein biomarkers, they can, the clinicians can advise a simpler genomic test, maybe ICH, basically ICH or a histopathology test, which is quite more affordable, cheaper, simpler, and in comparison to complex genomic tests. So based on this particular study, they have hypothesized that it can be classified into different therapy subcategories. So I think I would stop here, and now my colleague Abhishek will take over. And in the meantime, maybe we can have an open discussion regarding the same, whatever I have talked about so far.
Yeah, thank you, Kriti. Thank you for putting all this information together for us, for our audience. And I think we'll have the discussion towards the end when we have questions also from the audience. And
Sure.
Abhishek, yeah. So Abhishek, you can. Yeah.
So I'll just mute myself before Abhishek takes over. Yeah.
Hi guys, good evening everyone. Thank you for joining in on a working day, and thank you Dr. Angita for such a wonderful platform. I hope my voice is audible.
Yeah, we can hear you.
Sure. So thank you Kriti once again for such a informative and amazing way to bring genomics in cancer treatment at the very grassroots level. So I would take this discussion a notch deeper maybe, or expanding the horizon, if I may say, highlighting multiomics approach in liquid biopsy. So next slide, please. So as we know, and as Kriti rightly mentioned, that the easiest way go down to the happening in our blueprint, that is the DNA, is to do it through a sequencing. We take DNA, whether it's a circulating tumor DNA from the liquid biopsy. We do a panel, whether it's a Sanger sequencing or the targeted NGS panels, and we get the mutation. Now there are certain interesting things that we would want to know is, as Kriti correctly mentioned, that two patients, say of the similar age group, also being diagnosed with the same cancer subtype and automatically put into the same regimen. But why are they responding differently to the treatment? Maybe one person shows fantastic, recovery, where the other person shows a very delayed recovery, or maybe, it recovers, then pauses, and then there is a remission. Why does all of these things happen? The reason is that it is not just the changes in the DNA that are causing a problem. Yes, they are the source of the problem, but our human body is so complex that there are other players beyond the DNA who are coming into deciding whether the patient will respond to a drug or they will not respond to a drug. In addition to that, there are further complications in terms of, even our immunology, how our entire immunity is designed. That itself plays a very critical thing at where we see. So what often, at least, the adoption is very well valid in the US, the UK, and the global market, is to have a multi approach. In case of genomics, so one of the easiest, or if you say for example, all the drugs that are common in the country space, whether it's a drug, say for example, if I name a few, bevacizumab, they are all which target the protein cancer subtype. So even in your normal, for example, HPV vaccines, they all target a certain protein. The reason being, proteins are the players for any metabolic, non-metabolic, or any physiological process that happens in the body. They are blueprinted in the DNA, but ultimately it is the proteins that bring in the game or play the game body or health. It is also very important. Simultaneously, apart from studying the mutations or the changes in the DNA, we also know how the proteins are functioning in the patient body. Often there are technologies which help you to just have a non-invasive approach to study very targeted biomarkers, and the protein level helps you to give an early picture of how the onset of this mutation is happening. Well, the mutation onset happens early, mid-level, late, so on and so forth. The next slide, please. Genomics, can biomarkers help to detect in a blood test? Yes, as we can see, these are some of the examples where what they had done is that they have taken liquid biopsy. Yes, genomics, because next generation sequencing as a technology offers as that much of throughput to dive very deep into the molecular biology happening in the case of the disease with very limiting samples, high accuracy and precision. Itself is furthered by this proteomic analysis, where we study different biomarkers at that same level of precision. detect how the levels here, it's a case study where serum levels of LIF, which is a prognostic marker, actually is treated with ICV inhibition. So it shows how the levels of this marker corresponds to a particular inhibitory condition in the patients. So this gives us a very clear idea of whether the patient is suitable for a certain treatment regimen. Next slide, please. Again, so as Kriti mentioned, the tumor is doesn't stay in silo or in isolation in our body, right? So there are tumor cells, there are healthy cells, then there is the entire micro environment where we have immune cells, we have signaling molecules, and even within the tumor we have heterogeneity. So a tumor is not identically mutated cells. The cells have different shades of mutation, different grades of mutation, different statuses of mutation. So if we have a more targeted approach, then we need to not only identify the mutations but also see beyond the levels. Like if this mutation is there in the DNA, then what protein is getting impacted? So say a protein A is getting impacted. Now if the protein A is getting impacted, to what extent it is getting impacted? Does it become 100 to 0? Does it become 100 to 70? Does it become 100 to 30? Now if it reduces in a gradual manner, we can definitely try to understand that whether if a protein becomes from 100 to 70, is that an indication of onset? So that gives us a very good early understanding, and possibly the earlier we detect, the better is our chances of corrective intervention. Next slide, please. So this is a slide which is very dear to me because it perfectly encapsulates how the human biology or the human disease situation is. We have the blueprint in genomics, which is our DNA. We have the upgrades of the blueprint in the form of epigenomics, which Kriti touched upon. We have a Xerox copy of the blueprint, say transcriptomics, which is at the level of the RNA. And finally, we have the mediators, which are in the form of the proteins. It could be enzymes, it could be peptides, it could be proteins, it could be proteinaceous hormones. And then finally, most of these proteins work together to create even those smaller complex organic molecules, which we call as metabolites, which also impacts the functionality of our organs, of our entire systems biology to a great extent. So all these different aspects, when studied together, give us the complete picture of how the human biology works. Next slide, please. So as we see that in 21st century healthcare, what has happened is that with the advancement of science and technology, number one, we have better treatment regimens. Number two, we have better scientific tools to deep dive into the root causes of these different health conditions or diseases, especially cancer. Third is also there is a greater chance of their adoption into diagnostics and care or patient care. So as we see that from DNA to RNA to proteins, our scope gradually increases multifold. Each of them have a very significant role to play. Each of them have a very significant role to play in the disease prognosis and the disease treatment, and all of them together help in better treatment care. Next slide, please. So this is one of the reasons why the search of effective therapeutic targets become all the more complex. As I said, like just for example, think about HPV and the cervical cancer, and which is so common in India and globally, and the statistics are appalling. So it is not just one or two HPV. Right now, today we have two known HPV variants, as well as there are more than 10 high risk HPV variants that can trigger your onset of the cancer. And just imagine, each of these variants have a very different immune interaction with the patient body. So the immune reaction, the variation in the same family of viruses and their crosstalk together make the disease more complicated. So naturally, when I'm looking at such a complex condition, definitely our approach, we should have all the tools on the deck or all the cards on the deck that is possible. So that is where multi-omic approach is a very good fit in 21st century diagnostics and healthcare. Next slide, please. So one of the challenges that have been with traditional proteomics is the abundance of samples. We have sensitivity issues, specificity issues, sample consumption issues, and also the dynamic range, because it is very important to understand the significant changes in those protein markers at a low level. But now with the advent of genomics and proteomics and the amalgamation or marriage of them, we have been able to significantly overcome that. Next slide. Where using one of the proximity extension assay technologies, which is done on the NGS platform, we can detect changes in protein biomarkers at even femtogram levels. And we just need 1 microliter of patient sample. So which is amazing because that allows us the scope of extremely early detection of any disease onset. Next slide, please. So I will take you through two case studies. One is the next generation plasma proteomics, how it helps to identify suitable biomarkers for ovarian cancer. Now we know that apart from cervical cancer, breast cancer, ovarian cancers are one of the key problems or areas of cancer that affects women health. And as we know currently in the present scenario, it is a little difficult to distinguish between the benign and the malignant ones, and also the progress of the malignant tumors. Because in stage one, the treatment is really good, but the moment it converts into a stage two or stage three, the survival rate drastically reduces. So, what this group did was, they are based out of UK. Can I have the next slide? So what they did was that they did an exploratory approach of studying almost 1600 proteins and how their expressions significantly varied from benign to malignant tumors, as well as from early to late stages of the malignant tumors. And what they interestingly found was that they found about a panel of 4 to 7 proteins which are very unique changing in the progress of ovarian cancer. And therefore, now this panel has made it into diagnostics, which help them to distinguish between the benign and the malignant, as well as have an early detection of the onset of the cancer, so that the therapeutic intervention can be established. Next slide, please. Yeah, next slide. So one more such case is the non-small cell lung carcinoma, which is again a very aggressive form of lung carcinoma. Now, Anlotinib is a multi-target tyrosine kinase inhibitor, which significantly prolongs the survival, but somehow the response rate to this drug is really low. So therefore, it is very important to understand that which subcategory of patients should I treat this drug to, so that they are the effective ones who will be able to be managed by this drug, so that The other ones can be given an alternative mode of therapy. Next slide. So here again, an extensive study of the protein profiles of the protein biomarkers. They found that there are seven marker proteins which help to determine how the responders versus the non-responders profiles would be. So just imagine when a disease like cancer happens, it is such an emotional trauma for just not the person but also the family. And then of course they want to go all the guns blazing to get the best treatment, because the objective is to sustain and give a good life, a long life, and give a disease free survival. So all these multi-omics approach help us to understand how the complex the situation is and choose the best treatment regimen at onset, so that there are less trials and errors, and a more sure shot targeted treatment can be given to the patient, so that they respond early, they respond better, and they get disease free sooner. Thank you. That's all from my side.
Thank you so much, Dr. Abhishek. So I have a question for Kriti and Abhishek here. Since both of you are from the technology teams, I would a basic question that comes into the mind of clinicians while looking at genomics. First thing is that it is very expensive. How will we manage this for the patients? And what is the basic setup that they should have in their clinics to be able to make use of these genomic technologies?
Yeah, thanks, Ankita, for putting that up. So there are two ways that clinicians can do about it. So if funding is not a challenge, so definitely we need to establish a basic infrastructure in-house. So it all includes, first of all, the basic steps which are involved in the in the NGS test is you have to collect the sample, and then you have to extract the DNA or the RNA or whatever nucleic acid, whatever omic layers we are trying to understand, and then do a library prep followed by a sequencing. So if we have to establish an in-house genomic facility, we need to have, first of all, a good infrastructure, and some certain protocols at place, SOPs, for collecting the samples, just to make sure that we are not collecting any degraded samples. So we need to have certain SOPs. Infrastructure includes the cold chain and, for example, the -20 and the -80 freezers. Apart from that, a sequencing lab, which consists of a sequencer, and definitely once the data comes out, we need a perfect bioinformatics team in place. So in order to understand that data, in order to analyze it, and last step would be to better collaborate with a clinician to better interpret, basically a genetic counselor, as I mentioned before, or a molecular pathologist, to better understand the reporting. So one thing is that we can develop an in-house genomic facility, and all the infrastructure is required. We need trained professionals both for wet lab and for analysis bioinformatics, and then we should have certain collaborations in place. The second alternative is when the funding is a challenge, then generally what clinicians do is they collect the samples from the patients and they outsource it. And there are certain service providers in India which will get it done for you, and basically you will have to fund them, and they will get it done for you end to end, and they will give you a testing report which you can use for your reference to determine a personalized therapy.
Right, Kriti. So we have Professor Arvind with us today, and sir, I would like to ask you a few questions, because you are right in the clinic, from the clinic, and you deal with this day in, day out. So how do genomic alterations, for example, EGFR or BRCA, impact your treatment planning?
Yeah. Hi, Ankita. Is my voice audible?
Yes, yes, we can hear you.
This is the first time I'm using this platform, so I was a little unsure as to how it works. So I just had to download. And the other thing is my network okay? I'm slightly traveling, so there may be some sort of
Yeah,
signal drops occasionally. So pardon me for. Is it okay? Clear?
so far it's good. Yeah.
Yeah, yeah, great, great. And I think thank you for the opportunity and two wonderful lectures from two eminent speakers. I really enjoyed it. Of course, from the clinician's perspective, yes, I think we've come a long way. Like when I used to train, it was the one size fits all approach. No, we used to sort of throw everything at the patients. A lot of patients got over treated, and as a result, you had these side effects of treatment, sometimes futile treatments as well. But now I guess everything is going the precision way, and rightly so. And I, being a surgical oncologist, also my basic domains are to deal with some solid cancers. And as you rightly had asked the question about EGFR and BRCA, I think maybe lung cancers and breast cancers, and more so lung cancers, have been the prototype for driving precision medicine, essentially. So initially, we knew that how it all started was initially lung cancers was considered to be a smoker's disease, like 80 to 90% or more were associated with smoking. But over the years, globally and more so in India, we did find, and our group also find much earlier, maybe in early 2010s, we found that a lot of non-smoking associated lung cancers were there. And this was a finding that was seen in many other centers, contrary to what was seen at the start of the decade. At the start of the decade, we used to see more of smoking driven squamous cell carcinomas. But I guess in the next 10 years or so, we saw more and more adenocarcinomas. And now the trend broadly is that we see more and more of adenocarcinomas. More than 50 to 60% of our cancers, lung cancers or non-small cell lung cancers, are adenocarcinomas. A lot of them, we are seeing an increase in adenocarcinomas among non-smokers or never smokers and in women. And a lot of them seems to be driven by some mutations, more commonly, as you had mentioned, EGFR mutations. And in our subset, or at least the Indian subset, we find close to, and the proportion of lung cancers with EGFR mutations also seem to vary. Whereas in the US, I guess the numbers were slightly lesser. In the Asian population, it was in the range of around 40 to 60%. For us, I guess it's in the range of 30%. But then now with having NGS sequencing available in-house, at least in the cohort of patients that we have been seeing, we are finding an EGFR rate of around 40% or so. Of course, most of our lung cancer patients are metastatic lung cancer patients, so you do have a lot of treatment therapeutic implications, because you'll have to incorporate some of these yeah, anti-EGFR therapies in upfront in the management of these patients. And that we have also finding that a lot of these patients have got a better prognosis and outcomes are slightly better. With regards to BRCA, of course, a lot of things have changed. Initially, of course, we believed that BRCA was only to sort of know about the presence of the mutate, about these deleterious mutations, and also to counsel families. But now increasingly we are finding a lot of therapeutic implications also with the availability of certain PARP inhibitors. So maybe I can just go on, but from the clinician perspective, a lot of sea changes have happened from the diagnostic perspective, personalized medicine perspective, and from the prognostic perspective, and all of them have been elegantly highlighted by both my previous speakers. Over.
That's great to know that clinics are actually utilizing these resources and making the lives of patients a little better. One more question, Professor Arvind, that in your opinion, what kind of genomic insights are most immediately actionable for clinicians to decide? Like what kind of tests or genomic things promptly
No,
tell you?
I
Yeah.
think we need to understand, and of course, nowadays most of the genomic reports come with a clinical information and summary. So I think it's more important for us to sort of understand how a basic NGS report is, and then we sort of try to sort of make sense of the whole thing essentially. So we need to so I think from the clinician's perspective, we need to understand the variant finding, what exactly is the variant, and more importantly, as was said, the variant annotation, and most importantly, the interpretation and actionability of it. And as a clinician, and more so also from the patient's perspective, it is important for us to sort of see if any of them um, have certain personalized targets, so that, and with good evidence. For example, EGFR, you have at least 7 or 8 different types of targets you can sort of or treatment options that you can have if the EGFR is there. And even with the ALK also, you have at least 3 or 4 different types of inhibitors. Now, of course, we are going for the third generation ALK inhibitors in the treatment parlance. So I think more so from the patient's perspective, I think if some of these mutations have a targetable drug, I think that seems to make the greatest impact. Of course, from the prognostic perspective also, it's very good, so that you can sort of tailor make, or you can talk to the families, monitor also how you follow up these patients, and a lot of other different things also, how you sort of detect recurrences, and all those things can be tailor made, that follow up. And maybe in certain cases, of course, not much in solid tumors, but the significance of MRDs in these cancers as well. Yeah, over.
Absolutely. What you say totally makes sense, Professor Arvind. Thank you so much. Thank you so much, everyone. And now we open the floor to questions for the audience. So if anyone has any question, can please put them in the chat section. So there is a question that, can you share a case where genomics changed your clinical approach, Professor Arvind? This is.
Oh, many cases. In fact, a lot of in fact, as a matter of rule, all our cases of lung cancers, like would go the genomic way essentially, and preferably unless and until they are symptomatic. If they are symptomatic for some other reason, maybe we would start a treatment for them. But by and large, for patients, we try to sort of wait for these wait for the results of the sequencing, and then only we start a treatment for all these patients. There are some patients in whom there is multiple metastatic disease, in which you don't really have known primary, like the patients have multiple skeletal metastasis, brain metastasis, and many other metastases, and and sometimes it just the biopsy report is adenocarcinoma, and you don't really know where they say it could be from the breast, it could be from the lung, it could be from the GI. So in all these patients, I guess the sequencing has really helped us to zero in on a potential primary, and sort of give targeted treatment for some of these patients. I think I
Can't hear you, Professor Arvind. Yeah, lost you for a while. I think the signal is gone because he's traveling.
Yes, I believe we have lost it.
Yes. So no worries. Before we we'll wrap up the session now. Before we wrap up, I would like to extend a sincere thanks to our expert speakers for generously sharing their time and their valuable insights with us, the team at Primas Life Sciences for their collaboration, and to all of our participants who took out time on a working day to be a part of this schedule. So this was just episode 1 of our journey together. Stay tuned as we continue the series with more in-depth discussions. And for updates and upcoming events, you can follow ICGO on LinkedIn and check our website. So until next time, keep learning, keep collaborating, and let's move closer to personalized precision care in India. Thank you and have a good evening. Bye-bye.
Thank you so much.
Sankita, thanks for having us here. I hope we were able to make a suitable addition.
Definitely.
Yes.
Yeah.
I'm
Thank
sure we
you.
could teach your interest. And
Thank you so much, everyone.
Thank
Bye.
The recording has been stopped. Thank you for joining. Goodbye.